so much for coming, to those of you in person and those on Zoom, for Anna Heasley's thesis defense. I am really excited to get to introduce Anna today. Yeah, I don't want to take too long because we have a lot to learn about population genetics and relatedness of Olympia oysters. But before we talk about the familial relationships of oysters in captivity, let's talk a little bit about the student who did this work. So this is Anna. You could go ahead and advance the slide, Anna. So Anna came to Moss Landing Marine Labs from Pennsylvania, where she did her undergraduate degree in biology at Slippery Rock University. And growing up in Pennsylvania, she didn't have a coastline, and yet still Anna was a scuba diver before she even came to Moss Landing Marine Labs. So here she is getting her dives in while working toward her dives needed for her scientific diving. And so when she applied to come to Moss Landing Marine Labs for a master's, she was interested in potentially doing a project that might involve some field work and some field ecology or some physiology. But that said, you know, she had visited the California coast with her family, but it was a big transition to then move out to the marine environment. And so, next slide. Anna made a big embrace of living on the coast. And so this picture, Anna shared this one with me on the right. This is her tide pooling for the first time in Monterey. And she was always a great tide pooler when we would go out for class and when we'd go out for lab activities as well. She also brought her parents to the beach and tide pooling when they visited. And I think just generally, it was always a joy to go out with Diana. With Anna, because she has a knack for like dissection and for finding things. And so she was always really great at, you know, even finding, especially bivalves. I remember one time a jingle clam, she dissected it right there on the beach and started identifying its anatomy. So a true invertebrate zoologist through and through. Anna didn't come to this coast alone. So she moved out here with Jake, her partner, and she and Jake got their first apartment here. He has been her buddy, both dive buddy. This is diving in the quarries in Pennsylvania, but also her partner and support out here. And so you'll see some science contributions that Jake made in the talk later today. And as Anna was here, her field ecology and ecologist inclinations were supplemented by some interests that she developed from classes here. So she enrolled in the molecular biology techniques class taught by Sarah Smith and me. Sarah is on her thesis committee. And she really embraced molecular biology and really loved the approaches, the lab work, and she really thrived at lab work to the point where then she took a job in John Geller's invertebrate zoology and molecular ecology lab and was a research technician in there. And her pipetting skills came in handy in other classes as well. So you can see her pipetting for her thesis on the left, and then I presume in class on the right, pipetting cultures. And so developing those lab skills has turned out to be a pivotal moment for Anna in her thesis. And she made the decision to embrace that fully and change her thesis approach to involve molecular biology. And I think that's a really exciting thing that you can do in a master's project, and your time as a master's student is exploring your interests and making those changes. And so with that, Anna pursued a molecular and a population genetic approach to studying the Olympia oysters, which she had come to study initially. That said, she developed plenty of other skills while here, a well-rounded Moss Landing education. Anna is very seaworthy, and so she went out with the invertebrate zoology class and other classes here. She and I shared stories of seasickness and seasickness remedies to varying effectiveness. So yeah, she really is a well-rounded field and lab biologist. She also is an excellent science communicator, as we will soon see, for her thesis defense. So during her time at Moss Landing Marine Labs, she presented at the Western Society of Naturalists conference and at the, ICRS. It was a shellfish, yes.
International Conference on Shellfish Restoration.
That was it. ICSR. And that
You're
was
so
just in
close.
2024. And got a lot of really great questions and a lot of interest in the research that she's doing. So it's really relevant for shellfish research generally, what she has done with her work today. She's also a great science communicator and teacher. So here in the middle is Anna at the Touch Tank at MLML's open house teaching the public about invertebrates. And she was also a fellow with the SJSU Civic Action Fellowship. And so as a fellow, she taught coding to underserved youth in San Jose. So really just a great teacher and researcher. And she had great support, which we always loved hearing about. And then as Anna moved back to Pennsylvania to wrap up the later parts of her thesis, we got to have Zoom visits by Sheila and Winnie, who were great supports, and some of the favorite vertebrates members of the Invertebrate Ecology Lab. So yeah, with that, I don't want to keep talking because I want us to hear about Anna's research and everything that she's achieved here. So I'm happy, let us all welcome Anna as she presents her research, It Shucks to be Inbred, awesome title, Effects of Small Population Sizes on Genomic Diversity of Hatchery-Reared Olympia Oysters.
So thank you so much, Amanda, for that lovely introduction. You make me sound so interesting and cool. Thank you. Okay, so you've already read my title, so let's just jump on in. So to orient us, I'm going to start with some background just to get us familiar with some of the terms and ideas that we will be using throughout this talk. Okay, so Olympia oysters. Olympia oysters are marine bivalves that live along the west coast of North America. And they are the west coast of North America's only native oyster species. So they're pretty important. And they are considered a small oyster species. So I've included this figure in the center here that shows just the differences in size between oysters. So Olympia oysters can grow to about 2.5 inches to 3 inches, compared to the Pacific oyster that can grow up to 10 inches in the wild. So they are pretty small. And they were once very abundant. And we know this by analyzing some of the old Native American middens that are present along the coast, and also from some primary sources in the literature. So what really marked, well, okay, so they once were very abundant, but have seriously declined over the years, mostly historically, but also in the modern times as well. So one of the major drivers of the Olympia oyster populational declines was overharvesting. And this happened really largely in the California gold rush era. And this is just an image of what it kind of looked like back in the day. But they were fishing oysters so they could support their growing, settling population. And also eating Olympia oysters was seen as a sign of good fortune. So they were highly sought after. And there are other factors that really do influence the distribution and abundance of Olympia oysters today. I'm just going to name a couple for time's sake, but water quality is a big one. So this table comes from a paper from Kirsten Wasson through the Elkhorn Slough. And I'm just going to highlight a couple of the parameters here. But what this table is showing is the relationship between different water quality parameters and where we see oysters and where we don't. So I'm going to draw your attention to nitrates, phosphates, and ammonias. So we see that in areas that have higher nitrates, phosphates, and ammonias, we see less oysters. And in areas where these abiotic factors aren't as prevalent, we have better water quality, we see more oysters available in abundance. And it's not really surprising that there is varied water quality along the coast of North America, especially in California. We have the Salinas Valley, which is a mecca for agriculture. So we do have agricultural runoff that can influence water quality. Another factor, and perhaps the biggest factor today, is increased sedimentation and largely habitat loss. So oysters, like Olympias, require a hard substrate to attach to grow, and ultimately reproduce. So with loss of habitat and also this increased sedimentation, we see less and less oysters. And I've included this figure here from that same paper from Kerstein. What it's showing is the relationship between sediment depth and where we see live oysters. So we know that oysters are filter feeders and they don't like to be submerged in mud. They have to filter the water to be able to feed. So they don't like to be submerged. So in areas that have really deep sediments, so really mucky, muddy water or benthos really, we see live oysters living on larger substrates. So they don't want to be down in the mud. And filtering out this sedimentation is really kind of energetically costly to the animal, so they don't like a lot of sedimentation. So given the historical and modern declines of Olympia oysters, there are several restoration efforts in effect today. And the Native Olympia Oyster Collaborative, or NUC, does a really, really great job at cataloging a lot of the restoration efforts that are ongoing. And this figure on the right shows some of those restoration sites that are working to really bolster Olympia oyster populations. And I pulled this from their interactive story map, and I do implore you to visit their site because they have some really great resources. So some of the restoration efforts that are ongoing include deploying artificial substrate. So we can see Moss Landing's very own Jacob Harris out in Elkhorn Slough. And what he's doing is deploying some of these shells that are suspended on stakes. And these shells are really great spaces for baby oysters to attach to grow and develop and ultimately remain in that system. We also have larger scale operations that include deploying these large concrete oyster balls out into the water to do the same thing, provide habitat for oysters to grow. And a lot of these restoration efforts, they're very important for sure, but they are more helpful in areas that have enough oysters out in the wild currently, because we need adult oysters to produce babies to ultimately increase recruitment in an area. So we turn to other strategies to bolster those wild populations. And one of these is conservation aquaculture. So taking oysters out of the wild, bringing them into an aquaculture facility to grow and reproduce. So we can raise these babies up in a controlled environment to then be released out into the wild. And then they could grow and reproduce, and their babies could settle on some of those substrates that have been deployed out into the wild. So Olympia oysters have a really interesting life history. They are sequential hermaphrodites, meaning that they change sexes throughout their lives. And they also have this really interesting brooding strategy. So we have male oysters that will release their sperm into the water column. And for this figure, green is pelagic. So we have those sperms in the water column, and the eggs will remain inside of the mantle cavity or inside of those shells of the females, where the mother will filter out the sperm, fertilize those eggs, all while inside of the mantle cavity. And then they will then develop into this trochophore larval stage. And it's got this little ciliated apical region. And this is kind of what it looks like. I love this video. This video comes from the Chilean oyster, Ostrea chilensis. So it is also a brooding oyster. And what we can see in this video are baby oysters being circulated through the mantle cavity of the female. And in here, she will sort them, she'll clean them. There's a ton of maternal care that goes into raising these oysters, these baby oysters. So following the development of the trochophore larval phase, the babies will be released into the water where they will become pelagic again and grow into this veliger larval stage where it starts to resemble what we expect an oyster to look like. It's got two shells. And then it will find a cozy place to settle, maybe on an oyster ball or a shell in the slough, and grow into this early umbo adolescent before then growing into that adult phase. So given the life history of Olympia oysters, there are some challenges that aquaculture managers do face with breeding this animal. And one of these we can't control genetic crosses. So we can't control which individuals actually fertilize each other. So some individuals may actually dominate a fertilization event. We might have one male that produces a ton of sperm while, all the other males produce very little. So that one male could ultimately fertilize all the females that are in a breeding program. So we end up with uneven genetic contributors. We also can't control for sex ratios. So Olympia oysters are sequential hermaphrodites, and we don't really know when they are female and when they are male. So this presents some challenges in aquaculture. I've included this little example here. If we have 100 oysters in our breeding program, at any given moment, we don't know how many are females or how many are males. And it could be that a greater number of those oysters are males at any given time. We could say 70% of them are males, and only 30% of those females would actually be contributing genetic material to the next generation. Along the same vein, we don't know how many are actually contributing. So again, if some individuals don't spawn, if we have some gametes that are from a few individuals dominating the fertilization, if sex ratios are skewed, we can get this condition where we have low effective population size, or how many, or just a small number of individuals actually contributing to the breeding event. And this can increase genetic drift and homozygosity and ultimately lead to inbreeding, just having decreased genetic variation, which makes the animal more vulnerable to disease, environmental change, and ultimately long-term decline. So these examining inbreeding in Olympia oyster aquaculture is definitely an important next step. Okay, so I've included this really great example. We all know and love dogs, but they are really a great example for the understanding the effects of inbreeding. So on the left here, we have the original bull terrier. It's got this elongated nose. It's a beautiful dog. And through breeding closely related individuals, we can concentrate desirable traits, which leads to what we see in today's animal. And this little diagram just shows kind of how dog breeders do this. So inbreeding can cause these really long contiguous sections of homozygous positions called runs of homozygosity. And I've included this little graphic here. We're going to introduce this soon, so we can talk about it later. So what we have are two parents, this parent generation up in here, and each has their own unique stretch of genetic material. We can see that the male has these orange bars and the female has this blue. So whenever they breed and produce this next generation of offspring, we can see that those offspring are inheriting some of that genetic material. Again, we see the orange and represented here on this individual and blue there. But what ends up happening is that the breeding of these two related individuals, so in this F1 group, we can inherit these really long stretches of overlapping segments. And these are called runs of homozygosity. And within these runs of homozygosity, they can harbor an excess of deleterious mutations that can form or that can lead to reduced fitness. And also over time, I will mention, runs of homozygosity with increased inbreeding will get very long, and they will also make up a higher percentage of the genome or the entire genetic makeup of the animal. So here's a real life example. This is also from dogs. So we see that in dog breeds that have high morbidity levels, we can see the relationship with the runs of homozygosity, the proportion of them that make up a genome, or this FROH value, or the inbreeding coefficient. So we see that in dog breeds that have very high morbidity levels, they also have very high FROH values compared to animals that have low morbidity levels and low FROH values. So we know that having some diversity in a population is very important for long-term survival and success. Okay, so bringing this back to aquaculture, this example comes from the Pacific oyster, Chrysostrea gigas. And what we can see is that in populations that are experiencing a lot of inbreeding, we have significantly less reproductive effort, hatching rate, and also less effective sperm, all of which would be very detrimental to an aquaculture operation if your aim is to bolster your reproductive output. Similarly, in the Pacific white shrimp, we see also less hatching rate or smaller hatching rates, less hatchery survival, and also pond survival. So it's really important that we start to examine these the risk of inbreeding and inbreeding in general in Olympia oyster aquaculture operations. So one way that we can really look at inbreeding, and it's very powerful, we can use genomics and take a genomics approach. And again, genomics is just a study of the entire genetic makeup of the animal. And genomics work for Olympia oysters has been done in the past. I'm highlighting an example here from Catherine Silliman in 2019. And she looked at population structure of wild Olympia oysters along the coast of North America. And these are some of her sampling sites. It was a huge study. It was a beautiful study. But what she did was take a genotyping by sequencing approach, the details of which aren't that important for our purposes here. And she identified these single nucleotide polymorphisms, or SNPs. And this diagram here kind of describes what a SNP is. So if we have an oyster that has DNA that is made up of these nucleotides, so your A's, T's, C's, and G's, we can see that for this oyster, we have an adenosine at this one region, while this oyster down here has a G at this position. So we call this a SNP. It's just a polymorphism. And also, it's important to mention that at the time, Catherine Silliman didn't have access to a high-quality reference genome, which really limited her sequencing approaches. But now, thanks to Kevin and Luke on my thesis committee and the Nature Conservancy for funding this work, we have a high-quality reference genome, which allows us to take a more sophisticated sequencing approach, which is what we did in this study. Okay, so what's this study about? My first question is genetic diversity impacted by captive breeding conditions? So ultimately, is there evidence of inbreeding in hatchery-reared Olympia oysters? And two, how genetically different are individuals across breeding programs? Really, to get at this question of whether or not we should supplement one breeding program with oysters from another to really support those breeding efforts and also increase genetic diversity to establish a really healthy line of oysters. So how do we do this? Okay, so for my project, I took a low coverage whole genome sequencing approach. And this diagram here describes it pretty well. So we can, for our example, these arrows represent oysters. So, and these tubes are oyster DNA. So we took oyster DNA from a ton of individuals and we sequenced it. And in a sequencing event, you sequence these small fragments of DNA, and these small fragments are reads. And with an approach like this, our depth of coverage, so how many times each fragment gets sequenced, is really low. And our aim for this study was 3 times coverage. And just to give you some reference, in human population studies, this coverage can be extremely huge, like 30 times or 80 times for some cancer genomics work. But this allows us to balance cost. So sequencing a few of these, sequencing the reads just a couple times really helps you balance cost. And also we're able to do this method because we have a reference genome available. So thanks again, Kevin and Luke. Okay, so I was able to sample from 3 separate breeding programs, with the focus really being on elkhorn slough. So again, thank you, Luke, and also thank you, Kerstin, from the Elkhorn Slough Research Reserve, for giving me access to those oysters. But I sampled from 3 separate generations: a parent generation, an F1, and an F2. And these values in this table represent my sample size. And interestingly, within this F2 group, I was able to sample some individuals that I'm calling wild. So those oysters were sourced from the slough, and it's unclear whether they came from animals that had been deployed out into the slough from these aquaculture operations, or if they were just natural recruits. And also just my captive group here, which I sourced from the aquaculture facility. So this allows us to do an interesting comparison later. Okay, next I sampled from Tomales Bay, from Hog Island Oyster Company. Thank you, Gary Fleener, the director of this operation. From this group, I sampled just an F1 group. And then finally, I sampled from this Morro Bay group. Thank you, Kevin, for giving me access to your oysters. And within this group, I sampled a parent generation and an F1 generation and got a total sample size of 279. It's also important to note that these 3 locations were identified as areas to implement conservation aquaculture to bolster those native population numbers. So I had to figure out a way to sample my oysters without killing them. If you know anything about oysters, they don't really want to open up. So we developed this anesthesia protocol, and we did this using magnesium sulfate and got some pretty great success, around 82%. And what this protocol really allowed us to do was relax those shells just enough for me to get inside of there and take a little snip of tissue. And the tissue that I snipped was mantle tissue, just around this margin of the shell. So I snipped the tissue and I stored my tissue at -20 until I was ready for DNA extraction, which I carried out using a D&EZ blood and tissue kit. They are cheap, easy, safe. And then I did some cleanup and I quantified my DNA as well before I sent it to the Roy J. Carver Biotech Center, or the CBC, for sequencing at that 3 times coverage. And this was done on a NovaSeq S4 flow cell. I've included this image because I just think they're so cool. That's the flow cell. Okay, so from this sequencing effort, I was handed a ton of raw data, over 6 billion reads, and our read target was 150 base pairs, and they were also paired. So it was a ton of data. And I also got this multi-QC report that described some of the quality of that sequencing run, which was of excellent quality, by the way. So once I was handed my raw data, I first paired my forward and reverse reads. So if we think back to that diagram of low coverage whole genome sequencing, those horizontal bars, each fragment gets sequenced in the forward and the reverse direction. So I first paired those. And then I trimmed the adapter sequences, which is represented in this like dark blue. And those sequences just allow the DNA to actually be sequenced on a sequencer. And then I trimmed my low quality reads, which is in red in this little diagram. Following this, I mapped those reads back to the reference genome, and I dropped samples that had mapping rates below 85%, just so we know that we were using quality data. And then I performed variant calling. So if we think back to this diagram of SNPs, I did this using ANGSD, so I did a few other analyses there, but what I did is identify these single nucleotide polymorphisms, or SNPs. And then I imputed missing data. So as just an artifact of low coverage whole genome sequencing, you're left with like some holes in your data with low confidence calls. So imputation uses an algorithm to infer the genotypes that are missing. And they do this using the reference genome, which is really interesting. So getting at my first question, is genetic diversity impacted by captive breeding conditions? Looking at the Elkhorn Slough group and across generations, that PF1, F2. And then I also looked at those parameters in the Tomales Bay and Morro Bay group. But what I did was use runs of homozygosity and also that inbreeding coefficient to examine inbreeding. So again, thinking back to this diagram, what I did was quantify the lengths of runs of homozygosity that were present in each of my groups. And I also calculated that FROH value, or the proportion of the genome that is consisting of a run of homozygosity. Both parameters will increase with the degree of inbreeding. So really long runs of homozygosity implies a recent inbreeding event, and the same is true for FROH. And then I calculated effective population size. So how many actual genetic contributors were there to each breeding event. And I did this using any estimator, and I chose 5,000 random SNPs to do this analysis. And it's important to note, I'm using this 5,500 rule to really interpret my data. So a population size of 50 has been recommended as a rule to really avoid inbreeding in the short term, while a population size of 500 is considered sufficient to retain evolutionary potential in a population in the long term. Okay, so really getting at my second question now, how genetically different are individuals across breeding programs? So first, I calculated FST, or the fixation index. And the fixation index is a measure of the genetic differentiation between different populations. And it's a value that ranges from 0 to 1, with values nearing 0 indicating very little or no genetic differentiation. So two populations with these values would be very, very similar. Compared to an FST value that's approaching 1, they are very, very different. And I did this using VCF tools. And I've also highlighted this, this measurement here. So this 0.25 threshold, this is actually used in human population genomic studies for distinguishing people from different ethnic backgrounds. So it's not really that different, but in these types of studies, that's considered a large genetic differentiation. And in addition to the fixation index, I also did a PCA, or a principal component analysis, which ultimately just reduces dimensions and helps you reveal patterns in variant data. And I did this using Planck. So what I wanted to do was plot my 3 populations and see how well they cluster together. If they don't cluster well, perhaps they are more mixed, and so on. Okay, so what did we find? Okay, from my sequencing efforts, if we think back, my aim was 3 times coverage. What we got was 2.19 times coverage, which isn't so bad. And after mapping those samples back to that reference genome and dropping samples with mapping rates below 85%, my sample size was reduced, and my table kind of shows that. So before filtering, I had 23 more obey individuals, and after I had 2. So all of my comparative analyses are between just that F1 group because I have representation from all 3 populations. But I also lost a couple individuals from the Elkhorn Slough F2 group, but not so many. And then I calculated the total genotyping rate, which we estimated at around 60%. Again, just as an artifact of low coverage whole genome sequencing, we do have some missing data, but after imputing that data, we were up to 100%. Okay, so the first set of results that I will be sharing really answer this first question: is genetic diversity impacted by captive breeding conditions, looking at ROH, FROH, and NE. Okay, so just to orient you to this plot, on the x-axis, we have our generations. So we have a parent generation, an F1 and an F2 for Elkhorn Slough. And on the y-axis, we have our runs of homozygosity lengths. So we can see across all generations, we don't have any significant differences in the lengths of the runs of homozygosity, indicated by these significant letters here. So they're all A, no significant difference. And they are also well below this 4 megabase pair significance threshold. And from the literature, this seems pretty standard. Runs of homozygosity that are nearing that length of 4 megabase pairs are indicative of recent inbreeding. However, what we're seeing is that our three generations aren't even close to that. So perhaps we're not in danger. And also, if our program is causing our oysters to become more and more inbred, we would expect this value to be significantly higher than the first aquaculture generation, which it's not. Okay, now looking at FROH, or that proportion of the genome that is a run of homozygosity, we can see again that they are far below that 10% threshold. Again, this came from the literature. That would be indicative of a recent inbreeding event. And what we see is that for each of these Elkhorn Slough generations, our FROH values are far below that. However, we do see some significant differences. So interestingly, the parent generation was not significantly different from the F1 or the F2, but we do see significant differences between this F1 and F2 group, again, indicated by these significance letters here. So this is pretty interesting, but it may be that we don't have enough like generational data to really establish a trend in this type of data. But again, we are so far below this 10% threshold that it may not be much of a concern for now. And just to orient you to what we see in like other studies, this example comes from a study looking at eastern oysters, and we can see that our values are kind of similar to these wild selected, or these wild groups in their study, and also some of their selected groups down here too. So in that 0 to 0.01 range. So our data kind of fits well into the literature. Okay, and then also if you remember, within that F2 group for Elkhorn Slough, I sampled from a wild population, so just what I collected from the wild. We don't know the origins, and then also from this aquaculture group. And what we can see is that there are no significant differences in the FROH. So on the y-axis here, we have that inbreeding, that genomic inbreeding coefficient, FROH. They are not significantly different. And also they are far below that 10% threshold. So what this indicates to me is that our breeding efforts aren't making a significant impact on the inbreeding levels in these populations, at least from what my data has shown. And I love this plot, maybe others don't, but what this plot is showing you is where runs of homozygosity are distributed across the genome. So on the x-axis, we have scaffolds, which for our purposes are synonymous with chromosomes. So these are just genomic positions. And on the y-axis, we have individuals separated by generation. So these red dots are for the parent generation, yellow for F1, blue for F2. And what we can see is where runs of homozygosity are kind of shared across those generations by these like vertical bars. And we can see that they're pretty consistent across all generations. But what makes a plot like this kind of interesting is we can see where runs of homozygosity are kind of starting to crop up from one generation to another. And I tried to find some good examples of this in this plot, but luckily for Olympia oyster aquaculture, I couldn't find that many. So here's one on the left. We have a run of homozygosity at this position on scaffold 1 that is represented in a parent individual. And within this F1 group, we only have one individual that has this run of homozygosity at this one region. However, whenever we get down to this F2 generation, we can see that more and more of those individuals have a run of homozygosity at that position. And the same is true for this example, but we don't actually see it represented in F1. Okay, so then looking at effective population size, or how many genetic contributors we have contributing to the next generation. So if we remember that 5,500 rule that I discussed previously, I've placed that 50 threshold, so a population size of at least 50, on this plot to just show you how our generations kind of compare to that. And what we can see is that for each generation, they are the effective population size is above that critical threshold. However, for F1, it's barely. But what we can see is that for these two generations that were that were produced from the aquaculture efforts, we can see that the effective population size is actually increasing. So that's really great news. We're getting closer in the F2 group to that 500 threshold for long-term success in the animal. Okay, so now question 2, how genetically different are individuals across breeding programs? I also analyzed the runs of homozygosity and FROH values and effective population size in my two other populations as well. So we'll start off with those comparisons first. Okay, so again, just to orient you, on the x-axis, we have our populations. So I have my Elkhorn Slough, my Morro Bay, and my Tomales Bay groups. And again, these are all F1 individuals only. And on the y-axis, we have those runs of homozygosity lengths. And what we can see from this plot is that the runs of homozygosity were not significantly longer in any particular group. So again, looking at these significance letters, they are not significantly different. And all mean runs of homozygosity lengths were less than or equal to 1 megabase pair. Again, far below that 4 megabase pair threshold. So we can see that for each of these generations, or each of these populations, the runs of homozygosity aren't really giving us cause for concern for inbreeding in any given group. However, when we look at the genomic inbreeding coefficient, or that FROH, again, the proportion of the genome that is a run of homozygosity across each of these populations, we can see that the Morro Bay group has significantly higher FROH values. However, they are all under that 10% threshold. So this data really suggests that the Morro Bay group could be a target in the future for increasing those population numbers that contribute to these breeding programs. And this could be done through supplementing the population. Okay, and it's perhaps not surprising that the Morro Bay group has a significantly higher FROH value, especially when we look at the distribution of the runs of homozygosity for this group. So on the top here, those were those Elkhorn Slough runs of homozygosity distributions. But what we can see in this Morro Bay group here in purple, we can see that some of these individuals have a ton of runs of homozygosity, and they almost kind of smear together a little bit. So that would obviously contribute to that FROH value. And from this plot, we can also see that a lot of the runs of homozygosity that are present in one population are also present in the other populations. You can see these in like vertical bars. It's pretty clear. And we can also see where runs of homozygosity are relatively absent along the genome for all groups included as well. One of which would be scaffold 3, scaffold 5, and also scaffold 8. Okay, and then looking at effective population size, we can see that for all populations included here, that we have that the effective population size is above that threshold of 50, again indicated by this red dashed line, with the highest number of contributors seen in the Tomales Bay group. And this perhaps isn't surprising because this group does come from a more of a commercial aquaculture program, and they also have a lot more wild individuals in their natural populations. So, okay, now looking at genetic differentiation. So how different are these individuals from different breeding programs? My question 2. So this plot shows my PCA for all of my populations here. And what we can see is that individuals from Tomales Bay group pretty well with individuals from Tomales Bay. And the same is true for the Morro Bay group and also the Elkhorn Slough group. But what's interesting is that FST value, the FST values are pretty low across all comparisons here. And they do correlate pretty closely with geographic distance. So we see the largest genetic differentiation between the two populations that are the furthest apart geographically, which is interesting. So 0.04478. And we also see the smallest FST values between Elkhorn Slough, smallest, and Elkhorn Slough and Morrow. It sits right at that midpoint, so that makes sense. But all of those FST values they do fall within that little to no genetic differentiation range. So even though we do see some clustering in here, genetically, the animals aren't very different. So we could actually consider mixing populations in the future. Okay, so what are our takeaways here? For question 1, is genetic diversity impacted by captive breeding conditions? Again, I looked at the Elkhorn Slough, those three generations. And what my data suggests is that there is no significant evidence of inbreeding that has occurred across just 2 generations of captive breeding, so that F1 and F2. We also see that the effective population size is above that critical threshold of 50, which is great. So, and then we also, for my second question, excuse me, how genetically different are individuals across breeding programs, looking at all of the populations. My data suggests that all of my examined captive breeding programs do not display significant signs of inbreeding. And that also, given those FST values, populations are pretty well mixed genetically. Okay, so some future directions that we should take. We should definitely consider continued generational monitoring. And we should also, just by increasing the number of generations that we include in studies like this, and then hopefully we could establish a clear generational trend to further understand the impacts of our breeding programs. And also, we should consider mixing captive populations to really prioritize increasing that effective population size. So getting as many genetic contributors actually contributing to any given breeding program, ultimately to increase genetic diversity. So we can try to establish a really healthy natural population that we could, let live in these systems for years and years to come. Okay, so I have plenty of people to thank. I'm first going to thank my funders and my collaborators. So the California Sea Grant that directly funded me, and the California Ocean Protection Council, Prop 68, that funded the research, funded my research. So all the sequencing and everything. And also the Nature Conservancy for sequencing that Olympia oyster reference genome. And then all of my scholarships as well. My thesis committee, Amanda, Luke, Kevin, Sarah, and John. Without you guys, I couldn't have done this. And thank you so much, Amanda and Sarah, for teaching me in your class and getting me really interested in doing genomics work. And also to John for letting me work in his lab and really refine some of my molecular skills. And also for Kevin and Luke, all of the effort that you've made meeting with me on so many occasions, and all of your expertise and insights. Thank you so much. And I really want to thank Kirstine Wasson at the Elkhorn Slough National Estuarine Research Reserve. She gave me access to her oysters. She showed me around the slough. She encouraged me at some of my talks. She's just an amazing woman and an amazing researcher. So thank you. Gary Fleener from Hog Island, Phil Heller from SJSU. He got me set up on SJSU's high performance computing cluster and answered some of my analysis questions that I had early on that really helped me. Also, the CBC, the Roy J. Carver Biotech Center for sequencing my data, my invertebrate ecology lab members, I'm just going to name a couple, Jacob, Sydney, Keenan, Celine, Jackson, and all of the current members. Olivia Pollock for always being my friend and just being an amazing person. She came to my wedding, and whenever I was at Moss Landing and we started really hanging out, she pulled me out of the darkest time ever. So thank you. Also, the invertebrate zoology lab members for helping me with my lab work, Linda, Emmett, Katie, Felicia, and Alex. the MLML Aquaculture Lab, Ava and Andrew, Tara Eggink and Diana Steller, both for encouraging me and giving me pep talks whenever I needed them, and just supporting me as a grad student. MLML facilities, and also last but not least, the University of Pittsburgh and all of my current lab members or lab mates, Mary, Maureen, and Sydney. And you can see a lot of these people in this this little collage here. I want to thank my parents for always supporting me and just, giving me so much love and support. Olympia at my wedding, my cousin Rachel, all of my cousins, my aunts and uncles, my pit team here, Jacob, Sydney, Katie. And last but certainly not least, Jake, my husband. He has always made me laugh. He is just this beacon of strength and support. And he's come along with me on so many of my adventures in life, and I thank him so deeply. I honestly think he deserves this degree more than me, because without him, I probably wouldn't have completed this. So with that, I will take any questions that you may have.
Okay, thank you so much, Anna. That was fantastic. I'm happy to field questions in the room. You guys can talk directly at the owl as you ask your question. And we'll also, for those of you on Zoom, feel free to either use the raise hand function or type your question into the chat. I'm not sure that I can read those. I'm gonna drag them over here. Oh no, there we go. Okay, any questions in the room?
I have a question.
Yeah, Jess has a question.
Can you hear me, Anna? Okay.
Yes, I can hear you, Jess.
Okay, great job. I was noticing that Morro Bay, I think, had I can't remember which value it was, but it was like a higher value for some of those ROH ones. And I was curious if you thought that was because your sample size was lower, or if there's any other reason you think Morro Bay might be that way.
Oh, totally. So Morro Bay, their existing natural population of Olympias is very low compared to my other two populations. So it was estimated that there were none left in the wild. But recent studies have shown that there are about 500 left in the wild. So they do have a really reduced set of genetic diversity, I guess, represented in that group. So I do think it is more of a representation of the population than so much of my sampling.
Thank you.
And compared to the other populations too, the Tomales Bay group, that wild population is pretty big, estimated at under 1 million. So they have a lot more individuals in their population, more than Elkhorn Slough too, but Elkhorn Slough has more than Morro Bay.
Cool. All right, we've got a question from John in the chat. John Geller, he asks, why are runs of homozygosity positionally similar in the different populations? Do you think they're recombination hotspots?
Ooh, they could be recombination hotspots. So a lot of these runs of homozygosity are really short. So the ones that we saw on those plots are really short, and short runs of homozygosity are indicative of ancestral inbreeding. So this could have come from the ancestral group of Olympia oysters that kind of perpetuated those populations. So that's a really good question. I don't exactly know, but that's kind of my thoughts on that.
Yeah. Okay, I think I want to check the hand raised too. Okay, no hands on there. Okay, I have a question. I'm wondering if you were contacted by California Department of Fish and Wildlife or Nuke, and they were asking you to give some suggestions or recommendations about the varied approaches for conservation or restoration of Olympia oyster populations. when would you suggest, like, I guess, what would you suggest from your study in terms of aquaculture, mixing it with outplanting, how much you should mix your brood stock populations? Like, what would the recommendations be?
Yeah, so I think for certain systems where we have a lot of Olympias kind of naturally existing, focusing on those restoration efforts that I showed in the beginning, just deploying those artificial substrates out into the water, I think that's a really great strategy. It certainly has a lot less like human input, so a lot less effort for the humans. But I think in populations that have very few individuals left in their systems, we should definitely incorporate those conservation aquaculture strategies. And really, if there aren't a ton of oysters in your system and you have to turn to these strategies, my recommendation would be to supplement those populations with oysters from other breeding programs that are maybe similar enough that can be used as broodstock for that growing aquaculture operation. And the aim really is to have a sufficient number of genetic contributors in any given breeding event or brooding strategy, or yeah, breeding event. So just having as many oysters as we can, definitely above that 50 threshold. We know that if we have 50 oysters in a tank that we're trying to breed here, not all 50 are going to be contributing. So definitely getting as many oysters as you can in your program would benefit your program in the long run.
Yeah, that makes a lot of sense. Thank you. All right. Any final questions for Anna? Then I think we should congratulate Anna for a great talk. And for those of you on Zoom, feel free to pop up your screens if you want to give any congratulatory remarks in the chat. I realize we're just going to like whisk Anna away to another Zoom call. So feel free to take a couple minutes here to chat before we hop into that call.
Thank you.
To those of you in the room, there's refreshments. Please feel free to grab and celebrate Anna. Sorry we didn't DoorDash some to you, Anna.
Okay.
You've got a plant in the mail. That's